Okay, so we'll start the session, and I think students will keep on joining for 10 to 15 more minutes, but that's fine. We'll start. Okay, let me just, yeah. Okay, so this week we have to study about dosage, compensation, and X chromosome inactivation. So, we have been studying about RNA, different properties of RNA, different types of post-transcriptional processing that happens. In the last class, we have studied about the different kinds of decay mechanisms that happens in RNA. If the RNA is not good, if the RNA is premature, then how the RNA is degraded. There are many different mechanisms like non-stop decay, no-go decay, etc. that we have studied. And we have studied a little bit about non-coding RNAs. So non-coding RNAs, we have essentially studied that these are the RNAs that does not code for any amino acid. That means they are not coding for any amino acid and ultimately protein. That is why they are known as non-coding, because they are not coding. But these non-coding RNAs earlier were known as junk DNA, junk genomic regions, which do not have any function. But now with uh, research, and now with all the latest studies and findings, we know that these non-coding RNAs have a lot of important roles in maintenance and processing. And one of the many important roles of non-coding RNA is in X inactivation and dosage compensation. Okay, and that is what we are going to study in today's lecture. So, non-coding RNAs can be of two types: normal non-coding RNA and long non-coding RNAs. So, long non-coding RNAs are the RNAs which are more than 200 nucleotides in length. That means they are longer. The sequence is long, and that is why they are known as long non-coding RNA. So, here we will be focusing mostly on the long non-coding RNA. In short, we call it as LNC RNA. Okay, so in this week we have five lectures. So we'll try to study the basics, just like we do in our all classes. Then we'll try to focus on the logic and the concepts, and then extra information you can always go and see through the PDFs. Okay. So our first chapter is about signal recognition particles and different modes of compensation. So basically, signal recognition particle is nothing but it is composed of proteins and RNA both. And you can see the structure of a signal recognition particle. Just a minute. Yeah. So you can see the basic structure of a signal recognition particle. It has protein components as well as RNA components. And RNA holds together the protein subunits of signal recognition particle, as well as it helps the signal recognition particle to bind to the ribosome. So this is in continuation with what we were studying, I think, earlier. Now, important thing to study before coming to this X chromosome inactivation and dosage compensation is tmRNA. Just a minute, I think someone wants to join. So like I said, for 5-10 minutes, I have to switch the screen frequently because students do not come at time. Fine. So tmRNA, as the name suggests, it's a hybrid molecule. Okay, tmRNA is a hybrid molecule which is half tRNA and half mRNA. Okay, so this tmRNA is a hybrid molecule which is half tRNA and half mRNA. So what is the function of this tmRNA? So tmRNA helps to rescue ribosomes bound to mRNA which lack the termination codon. Suppose if during translation, so ribosomes are attached to the mRNA, and that helps to in the translation of in the translation to produce proteins. We all know that ribosomes helps in the translation process. So ribosomes are released when there is a what tell you, actually you tell me, what causes the release of ribosome? What are the signals that leads to the release of ribosome from the mRNA? Can anybody tell me what kind of signal is necessary for the ribosome to get released from the mRNA and to stop the translation process? Nobody knows the answer? Anything, you can just give a try. Any random guess. Okay. So Kavita has said stop codon, and this is absolutely correct. So stop codons tells the ribosome, because codons is only telling the ribosome which amino acid it has to bring. If it is AUG, it will bring methionine and so on. So if there's a stop codon, so stop codon is a signal that tells ribosome that acha, now that the protein has protein is completely made, now it's time to stop the translation process. But what happens if in an mRNA termination codon is not present, or by some default the termination codon is removed, or there is any mutation in the termination codon? Then what will happen? Then ribosome is not able to get released from the mRNA. Okay. So if there is no termination codon, ribosome will not get any signal, and then ribosome will not be able to release from the mRNA. So in that situations, this tmRNA comes into picture. tmRNA comes like a hero and said that I will rescue ribosomes from the mRNA. And not only that, it helps in rescuing the ribosomes and the release of ribosome from the mRNA. It also adds a degradation signal to the nascent protein. That means, for example, if I'll just show you in another slide. I think someone wants to join first. Let me, yeah, fine. Okay. So suppose if this is your mRNA, and here should be the stop codon. Ribosome is here and it should stop here. But because there is no stop codon, so ribosome is keep on translating. So this much portion is extra. The default protein, the normal protein should be only of this much length. And there should be the stop codon and the process should stop. But this much portion, it is extra synthesized. This is extra. So this has to be degraded. Why this has to be degraded? Because this is not the original protein sequence. The original protein sequence lies only up till here. So that is what is written over here, that it not only helps in removing the ribosome from the mRNA, it also adds a degradation signal, so that extra, whatever extra or whatever unwanted sequence that has been synthesized, it is removed. So let's see how it happens. So you can see, I'll just do the slideshow for this figure. Yes, so you can see over here. Mm, I think, yeah. So you can see this is the normal translation process. So you have a stop codon, and then your, what is ERF1? It is eukaryotic release factor. So when the stop codon is there, it gets a signal, and then this eukaryotic release factors along with GPT comes, which helps in the release of the ribosomes. But if there is no stop codon, then these signals will come, but they are not able to release the ribosome. Okay, so that is what is written over here, that if the termination codon by some mistake is not reached, the ribosome gets stuck upon reaching the N3' end of RNA and has to be rescued. Since the stop codon is not reached, the newly made protein is probably wrong and needs to be degraded. That is what I was telling. Because the stop codon is not reached, so the protein it's not a normal protein. It's not a natural protein that can be used in the body, because it has some problem. So that's why this particular protein needs to be degraded. So that is why tmRNA, it helps in the degradation also, it helps in rescuing the ribosome also. So how it does is, so tmRNA, it has a portion of tRNA also, and it has a portion of mRNA also. So the tRNA portion of the tmRNA carries an amino acid alanine, whereas mRNA has a bunch of codons and also has stop codons. So what it does is that the tRNA portion adds an alanine. You can see this is alanine to the growing polypeptide chain. You can see this is your tmRNA. This whole is your tmRNA. From here, this is your tmRNA. Okay. Now this black portion is the tRNA portion, and this red portion is the mRNA portion. So this tRNA portion will add a alanine to the growing polypeptide chain. Okay. The mRNA enters the ribosome and synthesis of polypeptide is continued with the aid of normal tRNA until the termination codon is reached. So basically what will happen, that first it will this tmRNA, it will synthesize few amino acids. So it will first provide alanine, and like a normal translation, it will keep on synthesizing, keep on increasing one or two amino acids, and then eventually this mRNA portion of this tmRNA has stop codons. So stop codons would be provided through the mRNA portion. Okay. So this mRNA will help in providing the stop codon, and this tRNA portion of the tmRNA, it helps in synthesizing. I mean, this tRNA portion in a way kind of replicates the normal translation process. Then in the end, what will happen? Ribosome is released and the newly made fusion protein is degraded due to the signal sequence in the C terminus. Then what will happen? In the end, when what will happen? This mRNA will provide the stop codons. When stop codons would be provided, these release factors are able to release the ribosome from the growing mRNA chain. So now your ribosome is released. But what about the protein that has been synthesized? This protein is a wrong protein because it's a fusion protein, because it has some of its own sequences and some of the sequences which are added with the help of this mRNA. So ultimately this protein is degraded. So tmRNA, it helps in the degradation of the protein as well. So if I can say in short that tmRNA, it helps in the release of the ribosome. Okay. And next, what it is doing? It is also causing, sorry, just a minute. Yeah. Uh, So it also helps in protein degradation. Okay. So in summary, what we can say about this tmRNA? I'm just writing the summary for your understanding of this tmRNA. Basically, it is composed of tRNA plus mRNA. One point. Second point is that it helps in the release of the ribosome. Okay. And third point is it also degrades the protein. And important is that ribosome is released only when there is a missing of stop codon. Only when stop codon is not there, then only this release has to be supplied by this tmRNA. So is this clear to everybody? The concept of tmRNA, is it clear to everybody? Yes or no? Just let me know so that we can proceed for the next topic, that is X chromosome inactivation and dosage compensation. Okay. So today the attendance is very less. I think as we are going towards the completion of this course, few and fewer people are joining the lectures, but that's fine. So the 11 students, 10 students who have joined, so these are the students who are actually serious, it seems, because I can see some of the names who are there since class since first class. They are continuing, and they are consistently coming to each and every class, which is really appreciable. Okay, so now we'll come to the next portion, that is dosage compensation. So we have talked about non-coding RNAs. So non-coding RNAs can have some regulatory functions also. That also we have talked about in our previous class, if you remember. So the regulatory function of a non-coding RNA can either they can be transcriptional regulators, or they can be translational regulators, or they can be modulators of protein function, okay, or they can be the regulators of RNA and protein distribution. So these are the various regulatory functions of a non-coding RNA. One of the most important regulatory functions of an RNA, of a non-coding RNA, is dosage compensation. What is the meaning of this? Anybody who has a little bit idea? I think most of the students might have a little bit of idea about what is X chromosome in activation and what is dosage compensation. Anything, if you know, any basic idea that you have about this? Rukmini, do you know anything about this? So yeah, so Kavita is saying that equalize, and Janara is also saying that she has a little bit of idea. Okay, so equalize gene expression. Yes. So so um males have X and Y, and females have two X chromosomes. Okay, so the expression that is coming from this X and the expression that is coming from this X, if both of the X chromosomes are expressed, then it would be double. Here only one X is there, here two X's are there. That means the expression of the X chromosome has to be equalized between males and females. Okay, that means the level of expression should be equal. If the level of expression from X chromosome is, let's say, A, this is the level of expression from X chromosome, then here, since there are two X, it would be 2A. But this would create a problem, and this would create a difference in everything, because when the expression is different, the level of expression is different, then your metabolism, your phenotypic, everything would be different. So to equalize the expression of X chromosome between males and females, one of the X chromosome is inactivated. One of the X chromosome is inactivated. So to compensate for it, and this X chromosome inactivation is known as dosage compensation. Okay, so what is written, that in animals, males and females have different number of X chromosome. Yes, so to equalize the expression levels from X chromosomes in males and females, some sort of mechanism must exist, and this mechanism is known as dosage compensation. Let us see that how this dosage compensation takes place. So you can see there are three different species: mammals, C. elegans, and Drosophila. So now you see that in all the three species, this is your female, this is your female. In C. elegans, acha, this is male and this is female. Can anybody tell me what does this sign signifies? This sign, Anybody who can tell me what does this sign means? This is for males and this is for females. This means, okay, let me see if anybody has answered something. What does this sign signifies? This sign signifies male, this sign signifies females. This sign signifies, anybody who wants to give a try? Nobody knows? So when you study zoology, those species where they can exist as both male and female, what are they known as?
Hermaphrodite.
Yes. So these are hermaphrodite animals in C. elegans. So, so now you see that in all the cases there are two eggs. In across three species we are taking the examples. We are taking the examples of mammals, C. elegans and Drosophila. So in all the three cases, this X chromosome inactivation, or sorry, this dosage compensation is taking place. But the mechanism of this compensation is different across different species. How? Now we'll first see about mammals. You see there is one X, one Y, and this X is getting produced. What happens in the case of mammals is that one of the X chromosome is inactivated. One of the X chromosome is inactivated. It is removed. So now the expression from only one X is coming. Okay. So in mammals, what happens? X chromosome inactivation takes place. Now, in the case of C. elegans, this is male and this is a hermaphrodite which has both male and female. So that's why it is written as XX. Okay. So now what happens? That this will produce X and this means that the expression of both of the X is reduced. So there is a repression. So this would be equivalent to this. Now I'll just explain you with a simple maths logic. If this is 1 x gives, let us say 1 x gives 1 unit of expression. So this is 1 unit of expression and here also 1 unit of expression. Here there is 1 unit of expression, 1 unit of expression, but because one of the X chromosome is inactivated, so again we get 1 unit of expression. Here there is 1 unit of expression, again 1 unit of expression, 1 unit of expression, but what happens? That both of them are repressed. So now what will happen? This is 1 and this is 1 by 2 and this is 1 by 2. So ultimately this will also be equals to 1. So that means in the case of C. elegans, like in the case of mammals, what happens? That the expression of one of the chromosome is completely stopped. But here the expression of the chromosome is not completely stopped, but the expression of both of the chromosome is reduced to half. So now there is a repression of the expression of chromosomes of each chromosomes takes place. So earlier this chromosome should produce a expression of 1 unit, now it is producing half unit. Here also it should produce 1 unit, now it is producing half unit. So that means you can see from the symbols also, this is bigger X, this is smaller X. So both the X's are present but the expression is reduced. That is why it is called as repression. So there is repression of expression. This is your C. elegans case. Now let us see what is happening. This is 1, this is 1, this is 1. So ultimately what is happening? So this is also producing an expression of 1 unit. This is also producing an expression of 1 unit and this is also producing an expression of 1 unit. But this X chromosome gets hyperactivated. So now because it gets activated, it doubles its expression. So now it expresses twice the expression that it usually does. And here nothing happens. 1 plus 1. So here it is getting hyperactivated, here it is getting repressed, and here one of the X chromosome is getting inactivated. So this is how the dosage compensation takes place. Different species have different mechanism of compensation of the expression. Is this clear to everybody? The three different mechanisms: inactivation, repression, and hyperactivation. Is this clear? Okay, just a minute. Okay. If this is clear, then let us solve 2 3 questions and then we will move to next lecture. So the first question is, dosage compensation helps to balance gene expression. True or false? I'll give you 30 seconds, 30 seconds to 1 minute to think, and then I will switch the screen. This is a very simple question. Dosage compensation helps to balance gene expression. True or false? Okay, let us see what students have answered, anything or not. So only one answer I can see. Okay, 2 answers I can see. Kavita, Janara, Kanchana, they all have said true. Others, Jyoti is also saying true. Okay. Others, what others think? Supraja, what do you think? It should be true or false? Jasleen, what do you think? It should be true or false? Okay, Yuvanjali is saying also true. Supraja is also saying true. Okay, so it is true. Just now we have studied that dosage compensation helps to balance the gene expression, because in females there are 2 X chromosomes, in males there is only 1 X chromosome. So to balance the expression of X chromosome, one of the X chromosome has to be inactivated in the case of mammals, and in the case of Drosophila and C elegans different mechanisms are there, but the overall logic is to compensate the expression. So the answer is true. Okay, now next question, easy. X chromosome inactivation is known as dominance, epistasis, dosage compensation, or complementation. Okay. Let us see what students have answered. Okay, fine. So we have a lot of. So Janara has answered C, Kavita has C, Jyoti, Trishala, Kanchana, Supraja. So everybody has answered correctly. So just we have studied that X chromosome inactivation is known as dosage compensation. C is the correct answer. Does anybody wants to add something in this question? Do you think this question is a complete question or something is missing in this question? So normally when we talk, we link X chromosome to X chromosome inactivation to dosage compensation. But just now we have studied some things. So do you think anything has should be added in this question? Some word should be added in this question. So X chromosome inactivation takes place in which species? Tell me. Mammals. So this question should specifically, X chromosome inactivation takes place in mammals only. So X chromosome inactivation is one of the mechanisms of dosage compensation. It is not the only mechanisms. So that is what we should that is why we should try to learn new things, that we know what is. I think before this class also many students, you all must know about what is X chromosome inactivation and what is dosage compensation. But now we know that dosage compensation not only takes place through X chromosome inactivation, there can be repression of X chromosome and hyperactivation of X chromosome. So specifically this X chromosome inactivation is a particular method which takes place in mammals. Now we know this. Okay, now we will come to the next chapter where we are talking about now, since we are mostly focused on this thing, mammals and the phenotype and the molecular events that takes place in mammals. So we will mostly talk about the X chromosome inactivation mechanism in our subsequent slides. But just we can see here whatever we have studied, that is written here, that to equalize the dosage of X chromosome, gene products by means of inactivating one of the female X chromosomes in mammals. So in mammals what happens, there is inactivation of one of the female X chromosomes, and this, the one chromosome which is inactivated, this is known as bar body. This is important. Okay, always remember this point, that the inactivated X chromosome, this inactivated X chromosome is known as bar body. Drosophila, what happens, the expression of X is double. You can see over here, hyperactivation takes place. So in Drosophila, what happens? There is double the expression of genes along the X. There is double the expression. And C. elegans, both of the X chromosomes are partially repressed. Now, I have written here half and half just to kind of show you, make you understand the concept. Okay, it is not always like both will be reduced to exactly half, and the repression of both of them would be equal only. Okay, it can be 0.75, 0.25, like this. It can differ. It can differ also. I'm just trying to tell you that whatever it comes, the it is repressed in such a manner that the expression of these two X chromosome would be equal to the expression of one chromosome. That is the logic, you should understand. It is not like it is exactly getting half and it is also exactly getting half. No, it can be different, but ultimately by combination, I mean by addition of the both expression, the expression would be equals to X, the bigger X. That is the logic. Now we'll see the X chromosome inactivation. So now always remember, like we talk about, that it is not only about doing a certain thing, it is also about maintaining that certain thing. For example, if you study and if you are in your graduation or you are in your post graduation, right? So in your first semester, someone got the highest marks and topped the class. So is that the end? No. If you are the topper of the class, you studied one time, you don't have to just stop it there. You have to maintain it. You have to maintain it through all your semesters. So you have to keep on studying, you have to keep on hustling, you have to keep on working hard, so that you can maintain the status that you have earned by securing the first position of your class. Or this is a positive example. Something, for example, if you for example, if somebody has a fracture in your hand. Okay, so you go to the doctor and doctor put a plaster. So you have to take care of your hand and you have to maintain your hand up till your plaster is released. Similar kind of mechanisms happens in our body also. So body also tries to not only create things, but also it tries to maintain the thing. So if the X chromosome is inactivated, now this inactivated X chromosome is still there in the body, right? That inactivated X chromosome is still present in the cells. So now it has to be maintained. And this maintenance of the X chromosome takes place. And this inactivated X chromosome is known as bar body. And this inactivated X chromosome is actually present as a dark condensed structure that is found in most of the female placental animals. Okay. So this inactivated X chromosome is maintained as this inactivated X chromosome is maintained as bar bodies, and which is present as a darkly stained and a condensed structure. Okay, this is how it is maintained. Fine. Now we'll go that how this silencing takes place. Up till now we have studied that. Okay, I think someone wants to join or something. Just, yeah. Okay, so now up till we have studied that. Okay, we have to maintain the expression level of X chromosomes. So one of the X has to be inactivated. And that inactivated X chromosome in females, it is present as bar bodies. And it is a darkly stained and a condensed material. Okay. Now the question comes that how this X chromosome is getting inactivated. Here comes the role of RNAs. So ultimately, what we are studying RNA biology, not right. We are not studying about. We are not studying here cellular physiology. We are studying RNA biology. So now comes how we can relate this X chromosome inactivation and the compensation with RNAs. So the point is that this X chromosome inactivation takes place with the help of a non-coding RNA, specifically a long non-coding RNA, which is known as ZIST. Uh, XIST. In whatever way you want to pronounce it, that's up to you. Okay, so this X chromosome inactivation takes place with the help of this ZIST non-coding RNA. Just now I told you, can anybody tell me the difference between a long non-coding RNA and a small non-coding RNA? I told in the starting of the class, in the beginning of the class I have told. Can anybody tell me the difference between a long non-coding RNA and a small non-coding RNA? Basic difference. Nobody knows? Uh, so we have just discussed that non-coding RNA, which is more than 200 nucleotides. If a non-coding RNA has more than 200 nucleotides, then that non-coding RNA is known as a long non-coding RNA. Now what happens that the X chromosome silencing is mediated by ZIST, which is a 16,000 nucleotide long non-coding RNA. See, long coding RNA means a non-coding RNA which is more than 200 nucleotides. It's a 16,000 nucleotides. It's a very long LNC RNA. Now what happens that this non-coding RNA recruited complex has one entry site in X chromosome corresponding to cyst gene itself. So what happens that this is the gene. This is the cyst gene. Then what happens? The RNA is produced from this gene. And this is your cyst non-coding RNA. So this cyst non-coding RNA goes to its gene itself and then it recruits some histone proteins. Okay. So is it clear that this cyst will produce? What I'll do is that I will see here. So I give more time in explaining the concepts and the logics, and then the other detailed information, the reading part that you can do, you have the PDFs available that you can do. But the logic, the concept, that needs to be cleared first. Basics are the most important thing. So if this is your whole sequence, and this particular is a cyst gene, it will produce LNC RNA, which is cyst LNC RNA. Now, this cyst LNC RNA, it will go back to its gene. It is recruited to its own gene. And then what happens? After getting recruited to its own gene, it brings some kinds of histones. Okay, some kinds of histone proteins comes. And then what happens? These histone proteins, this particular isoform is H2A 1.2. So this isoform H2A 1.2, it comes, and ultimately what happens? This maintains the chromosome in inactivated state. Okay, so this cyst non-coding RNA, it gets back to its own gene, and then it recruits some of the histones, and these histones maintains the chromosome in inactivated states. So this is how this cyst non-coding RNA helps in repression of the X chromosome. So it maintains the X chromosome in inactivated state. X chromosome, specifically the X chromosome. Okay, so what happens basically? This is one thing. Other thing is that cyst containing complex recruit histone deacetylase and methylase. Now always remember, it's a basic concept that you should always remember, that transcription, when we talk about transcription, okay, so whenever there is transcription, if there is methylation, I think I have discussed it also earlier. So methylation will activate transcription or it will repress transcription. Can anybody tell me? Methylation activates transcription or it represses transcription?
Press.
Repress transcription. Okay. And acetylation? So it represses transcription. And acetylation? It represses transcription or activates the
Activate.
activates the transcription. Good. So methylation repress transcription, and acetylation, it activates the transcription process. So what does it means? It means that if the transcription of X chromosome has to be stopped, there has to be increase of methylation and increase and decrease of deacetylation, increase of deacetylation. That means, because acetylation is increasing transcription, and the opposite of acetylation is deacetylation. So if both this deacetylation and methylations are increased, then the transcription would be inactivated. So this is what this cyst does, that it recruits some histone deacetylases and methylases, which deacetylates and methylates the histone proteins, and therefore the expression of X chromosome is stopped. One of the X chromosome is stopped. So this cyst act, is this clear? Up till here, is it clear how this cyst is silencing the expression of one of the X chromosome? Is it clear to everybody? Okay, fine. Now this is fine. We have studied that, okay, this cyst will produce this, the cysteine will produce a long non-coding RNA, and this long non-coding RNA will go back to its own gene, and by going back to its own gene, it will recruit some histones, and these histones maintains the chromosome in inactivated states. Alternatively, it also does what? That it also recruits complexes which deacetylate and methylate the histone proteins, and therefore this X chromosome is inactivated. But in cellular biology, when you study about cells, when you study about the complex networking that takes place inside the body, it is very interesting and rather surprising to see that each and every molecule is regulated. Each and every kind of complex process is so well and so intricate that everything is regulated. So we cannot even try to replicate the kind of complex mechanism that our body creates. So you see that X chromosome has to be inactivated, and to inactivate the X chromosome, there is a long non-coding RNA which we call as cyst. Now there is another long non-coding RNA which regulate this cyst. See, for X chromosome, cyst is there, and for cyst, there is another long non-coding RNA which is known as T6. Now you see here, This is cyst. And to control its activity, the antagonist molecule is, see, TSIX, just the opposite of it. So if it is an antagonist, what do you think this T6 will do? If it is an antagonist of cyst, what do you think would be the function of this T6? Can anybody tell me? If T6 is the antagonist of cyst molecule, then what would be its function? Think, what would be its function then? Its behavior is opposite to that of cyst. Antis Antisense, okay. Excuse me. Okay. Oh, sorry. Mm, ultimately, you should remember that it has an antisense activity. Absolutely correct, you are saying. So, in a general picture, if this cyst is reducing or inhibiting the expression, this T6 will activate the expression. Okay, because this is inhibiting the expression, and this T6 will activate the expression. Okay, this is like a general way. And how it will activate? It will activate the expression by regulating T-cyst. It will activate by inhibiting T6. Now, how it inhibits T6? Let us see. So, cyst activity is regulated by another 40,000 nucleotide long non-coding RNA, which is known as T6. Okay. So, what does it do? It contains an antisense sequence of cyst, and therefore it is able to regulate the cyst activity by base pairing to it. So, it base pairs with cyst, and then it regulates the activity of the cyst molecule. So, this is important. It has an antisense sequence of cyst, and therefore it able to regulate the cyst activity by base pairing to it. We'll see in detail about it. Let us move the mechanics. So whatever we have studied, just it is written in the words form. Let us just see. So cyst expression is followed by irreversible layer of chromosome modifications that includes the loss of histone, which is acetylation, and this methylation, and that are associated with active chromatin. So basically what happens, that when cyst is there, cyst comes. So the function of cyst is to inactivate the X chromosome. Okay, this is the function of this long coding RNA. So for the inactivation of this chromosome, it does a number of chromatin modifications, and these are the name of those chromatin modifications that it does. Hypoacetylation, because acetylation it increases the activation. Hypo means less, so acetylation is reduced, methylation is increased, hypermethylation, methylation is increased, and ubiquitination is also there. So basically all the acetylation events are inhibited and methylation events are increased. That is why it is written hypoacetylation and hypermethylation. Hypo means less, hyper means more. Just like for example, we have hyperthyroidism and hypothyroidism. So hypo means less and hyper means more. Okay, so one important thing that I wanted to tell you, that when we talk about chromatin modifications, so what do you think, what is the meaning of chromatin modifications? Can anybody tell me what is the meaning of chromatin modifications? Anybody who what you can, if you can guess anything, what is the meaning of chromatin modifications? Anybody wants to try, Vanaja? You have any idea about chromatin modifications? No? Okay, fine. So basically, yeah, anybody wanted to say anything? No? Fine. So, see, the chromatin, we know we have a euchromatin and a heterochromatin. So, which is the transcriptionally active? Euchromatin or heterochromatin?
You. You
Euchromatin. And which is darkly stained? Euchromatin or hetero?
Hetero.
Hetero. So, why you think that heterochromatin is darkly stained? Why?
It is inactive.
It is inactive, but why? What has led its staining to be so dark?
It
Because
is
it is
highly condensed.
yes, it is highly condensed and it is highly compacted structure. That means the chromosomes are so much intertwined with each other, they are so much compact to each other, that there is no space for the outside transcription factors and outside modifications to take place, because it is so well intertwined with each other. That means for the transcription to take place, the chromatin has to loosen up a little bit. So when it will loosen up a little bit, then it will have a space for the transcription factors to come and bind to it. Okay. So that means that chromatin modifications, in a way, it helps in loosening of the or the compaction of the chromosome. So methylations will make the chromosomes even more compact, and acetylation will loosen the chromosomes. This is one thing. Another thing is that we talk about that a promoter is there, an enhancer. We talk about promoters and enhancers. So this promoter, it acts on enhancers and it enhances the activity. Or sorry, this enhancer acts on the promoter, it's the other way around. This enhancer acts on the promoter and it enhances the activity of the promoter. But what happens if you see in a linear form, this promoter and this enhancers are located very far to each other? So how they will come together then? That means some kind of chromatin modifications takes place that brings these two particular regions, promoter and enhancers, into close proximity. What kinds of modifications? If this is your promoter and this is your enhancer, it will kind of form a loop. So promoter and enhancer will come together. So this looping takes place. Looping, this is another kind of chromatin modification. This is not given in your this thing lecture, but I'm just telling you because it helps you to bring I mean to build your concept. So what I'm trying to say is that X chromosome has to be inactivated, and for the inactivation of this X chromosome, this zist non-coding RNA comes. And when this zist non-coding RNA comes, it does certain kinds of modifications in the chromosome, in the chromatin regions, and those chromatin modifications leads to the inactivation of X chromosomes. How? Because these chromatin modifications are making the chromatin even more compact, and they're bringing the promoters and enhancers very far away from each other. Okay, is this clear to everybody? Up till here, is it clear? Yes or no? Okay, fine. So now we'll see some questions and then we'll move to the next chapter. Okay. So now we have already studied about this. So what is the class of zist RNA? Non-coding RNA, long non-coding RNA, shRNA or piRNA? I'll give 1 minute to think and then I will switch the screen. Okay. Let us see if anybody has answered. Okay. So Janara has answered B and Kavita has answered A. Okay. Anybody else? Jyoti, you want to answer? Kanchana, Yuvaranjani, Supraja, Vanaja. What do you think should be the answer? We have been studying about it all way long. Try to attempt. Nobody wants to attempt? Okay, fine. So, okay. So, cyst is a non-coding RNA, but here see, if options would be non-coding RNA, shRNA, and piRNA, then obviously we will choose the non-coding RNA option. But here, apart from non-coding RNA, LNC RNA is also given. And we know that ZIST is specifically a long non-coding RNA. So this is more specific description of ZIST. So the answer would be B. Is it clear why it would not be A and why be B? Because it is a non-coding RNA, but the special, it is a long non-coding RNA, so more specific. This answer B is more specific. Had it been only three options, A, C, and D, then we would have definitely picked the first one. But here, because even more specific option is given, that is why we have chosen LNC RNA. Okay, Kavita, is it clear to you why it is B and not A? Okay. Now, next question is, bar body is the active X chromosome, inactive X chromosome, active Y chromosome, and inactive Y chromosome. Okay, let us see. Uh, okay. Okay, so Janara has answered B, Kavita has answered B, Yuvaranjani has also answered B. Yes, so B is the correct answer. It is an inactivated X chromosome. So do we need one very basic question that I want to ask, that is there any need for Y chromosome inactivation also? Y chromosome inactivation. Do you think that there is a need of Y chromosome in activation? Yes or no? No. So why there is no need of Y chromosome in activation? Why there is no need of Y chromosome in inactivation? Do females have a Y chromosome?
No.
That means only one Y chromosome is already there. So there would be no increased expression of Y chromosome, just like in there is no YY, or there's only XY, and in females there is no Y. So that means there is need of only X chromosome in activation. Okay. Now we come to the next chapter, which is chapter number 3. So this is just an example of random X chromosome inactivation. So you should always remember this example is there to tell you that X chromosome inactivation, it happens randomly. It is not fixed that only this particular chromosome will be inactivated. The inactivation can be random. So this calico cats means, so if you see a cat which has both black and red color patches on it, then it is always a female cat. There is no nothing is needed, no test, nothing is needed. If you have a cat that has both red and black patches, then it is always a female cat. Why? And this cat which has both black and red patches, this cat is known as a calico cat. Now, why this red and black patch cat is a calico cat is always a female cat? You see here that in the color. So this is your coat color, body color. So body color red or orange and black. So this orange is always dominant over the red one. Okay, this orange color is dominant over the red color. So in males, y does not have any color. So if XY is there and this X is of orange, then it would be an orange male. XY, if this X is a black, so it would be a black male. Males are always XY, so there cannot be XX. So either it would be orange or it would be black. Here you see, if both are black, it would be black. Both are orange, it would be orange. But what will happen if one of the chromosome is coding for the orange color and the other is coding for a black color? Now typically your orange is what, dominant over black, but here the activation happens and sorry, inactivation happens, but this inactivation is random. So what does it means? That in some cells, this black color would be inactivated, so orange color would come. In other cells, this orange color would be inactivated, so this black will come. So ultimately, what will happen? That the cat will have both black and orange patches, because this X which is coding for orange and one X which codes for black. So if the inactivation was not random, then what would happen? Suppose if black is inactivated, then all the cells have orange X, then it would be completely orange. But because this inactivation is a random process, so randomly in some cells X not would be not there, in some cells black X would not be there. So that is why, because it's a random, some cells would have a red color, sorry, orange color, and some cells would have a black color. So your cat would be of a like this, which has both orange as well as black color. So this is what is said over here, that gene for coat color has two alleles, XB for orange coat and X smaller b for black coat. Now orange is dominant over black, therefore heterozygous females XBB. Now, this heterozygous female in normal condition, because orange is dominant over black, so in normal condition, if there is no inactivation, then the color of the cat should be orange. But because here inactivation takes place, and that too random inactivation. So what happens? The heterozygous cat shows color patches due to what? Due to random inactivation of X chromosome in different cells of the blastula. So different cells of the blastula, different X would be inactivated. In some cells, orange colored X would be inactivated. In some cells, black colored X would be inactivated. So in this way, it takes place. So is this clear to you? This example just tells that the inactivation of X chromosome, it is a random event and not a specific predetermined or pre-made event. Is it clear to everybody, this experiment?
Yes, ma'am.
Okay, fine. So now we'll move further. So this is just showing, okay. So this X chromosome inactivation is actually a very interesting topic, I think this is one of the interesting topics of this unit. If you see how this takes place, what is the molecular mechanism behind it, so a lot of research is also going on in on X chromosome inactivation, which is it's a really promising area of research. In fact, I think sir has also shared one or two papers. I have not mentioned here, but if you go through the lectures, and you will find some of the papers which shows the kind of research that is being made and that is being done on this dosage compensation and everything. Anyways, you see, this is your X chromosome. So the X chromosome inactivation takes place basically. There is a center which is known as X chromosome inactivation center, XIC, and you can see that there are different promoters over here. You can see P1 promoter is there, P2 promoter is there, P0 promoter is there. So first what happens? Choice. What is the meaning of choice? Choice of the promoter. Which promoter has to be used? P1 has to be used, P2 has to be used, or P0 has to be used. So first is your choice. Second is your counting. Counting means how many X chromosomes how many chromosomes have to be inactivated. So first thing is choice, choice of the promoter, whether it has to take P1, P2 and P0. Why this choice has to be made? Because these promoters are active, or these promoters are important in different stages of a life, or in different stages of in the cell, different like some are important in the embryos, some are important in adult life. So in a complete life cycle, the role of these different promoter is different. That is why first important thing is, for the inactivation you need to choose which promoter has to be used. After you have chosen which promoter has to be used, you have to count the number of X chromosomes that you want to what you want to inhibit, and then you do the inactivation, cis inactivation. So choice, counting and inactivation, this takes place. So you can see this is the X chromosome inactivation center, and here you can see there is the cyst has these promoters P1 and P2, and the opposite is T6, which has just the opposing role of cyst. Okay, now important is that there is imprinted and random X inactivation. What is the meaning of this imprinted and random X inactivation? Can anybody tell me anything if you know? So I want my class, I always say that I want my class to be interactive. I don't want it to be a one-way class that I am only speaking and you are, I don't know, listening or sleeping or doing anything, because online you really don't know what the students are doing. So I want it to be a two-way communication. So I keep on asking and I will keep on asking questions. I'm not judging you. I'm not trying to test your knowledge and intelligence. I'm just trying to have a healthy discussion. Okay. So anything that you can understand by this term imprinted and random X inactivation? Anything that you can think of? What is imprinted? What is the meaning of imprint? Anybody knows the meaning of literal meaning of imprint?
Silencing.
Okay. Silencing the you can say that in this context we can say that the pattern of silencing is imprinted. That means whatever the pattern of silencing is there in the parents, that pattern of silencing is inherited just like a fingerprint. It is inherited from the parent to the child, to the offspring. Okay. So we talked about random X inactivation. So random means there is no specific memory. There is no specific pattern to it. Anywhere it can happen. Like we studied this example. Here we saw that any of the X chromosome can be inactivated. So some cells of the blastula will have X red as inactivated, others will have black as inactivated. But imprinted X inactivation means that the offspring, it has a memory. It knows that what X chromosome it has to inactivate. So that is imprinted on it, like prints imprinted on it. So that is what it is written over here, that in trophectoderm and primitive endoderm of the blastocyst, X inactivation is subjected to imprinting of paternal X chromosome. That means up till the epiblast stage. Okay, up till the epiblast stage, the paternal, that is the X chromosome that is coming from the males, that is coming from the father, it is only inactivated. This memory is there. So it is not a random event that is taking place. It is a specific event, well planned and well known event, that up till the epiblast stage, the paternal X chromosome would be inactivated. And this is known as imprinting of paternal X chromosome. That means the X chromosome inactivation event, it is not random. In this case, it is not random. After the epiblast stage or in the epiblast stage, what happens? Then the X inactivation becomes random. Up till here, before epiblast, it is not random. It is fixed. Okay. So here we have a random X inactivation, and imprinted means it's fixed. Okay. So early cyst expression is exclusively of paternal origin. The maternal cyst allele is repressed until the morula stage. That is what is saying. Because if X is inactivated in the males, that means the cyst is expressed in the males and cyst is not expressed in females. That is the only meaning. Because for the inactivation of X chromosome, cyst has to be expressed. So if X of the male is getting is getting inactivated, that means the cyst of male is also inactivated up till the morula stage. So this is basically you can see the X chromosome inactivation can either be random, which is mostly the case, and in some cases it can also be fixed. For example, the imprinted X chromosome inactivation of the male allele, of the paternal allele. So this is what this. Okay, let me do one thing. This is the diagram. Okay, and this is its text. Okay, so what it is saying is, I told you now, first thing here is what? First thing is choice. Choice of what? Choice of promoter. Which promoter has to be chosen? So you can see that P1 and P2 promoters are used for transcription of stable cyst. This is used for the see this P1, P0, P0, P1 and P2. These are the promoters, and these promoters are used for the transcription of cyst gene, which will produce a cyst LNC RNA. So P1 and P2 will produce the stable cyst, and P0 will produce an unstable cyst. Okay, this is clear. This is what comes in choice. You have to choose. You have to choose which promoter you have to use. And hyperacetylation disappears upon differentiation. Okay, this is all, you just need to read about it, nothing much to explain. Just stabilization of cyst RNA might depends on the level of cyst RNA present. Okay, and hyperacetylation disappears upon differentiation. Okay, these are just the points to explain. Now this is just another way to show diagrammatically how this X chromosome inactivation actually takes place. You can see sorry, so you can see this is the cyst area. Cyst, it produces the long non-coding RNA, and this non-coding RNA which is produced, it in cis manner, it produces an inactive state in the whole chromosomal area, and then other histone proteins are also recruited, which causes the which causes the which decreases the acetylation of the area, because acetylation, it promotes the transcription process. So this is just a way to show that how the X chromosome inactivation takes place. It is just a diagrammatical representation of the X chromosome inactivation. Okay, so again this is little bit of theory. So what they are saying is T6. So up till now we have studied about cyst. So what we have studied about cyst? We have studied, okay, can anybody summarize what we have studied about cyst? XIST cyst. Anybody? One or two lines? What you know about cyst now? XIST cyst. So why zist is used? Zist is used to dash. Zist is used for, what is the use of zist long non-coding RNA?
Inactivation of exons.
Yes, inactivation of the one of the X chromosomes. So zist is used for the inactivation of one of the X chromosomes, and the function of zist is regulated by another long non-coding RNA, which is T6, just the opposite of zist. See, T-S-I-X. So what it does is that it has an antisense RNA, so it binds with the zist long non-coding RNA, and both of them then forms a double-stranded structure, and therefore zist cannot perform its function. So what is the function of T-cyst? It is transcribed from antisense strand. Okay, in undifferentiated cells, T6 is expressed on both of the X chromosomes. In both of the S, in differentiated, it is present in only one. Prevent upregulation of cyst on activated X chromosome and influences X chromosome choice and imprinting. So, what is the meaning of this? See, this is the activated X chromosome. So, this T6, if there are two X, one is inactivated, other is highly activated. I mean the normal activation. So the one which is activated, this P6 is present in this activated chromosome. Okay, but this activated chromosome, the presence of T6, it is only in the differentiated cells. In undifferentiated cells, it is present on both T6 and zist, and it is a matter of chance, whatever will be encountered, whether zist or T6, that will do its function. We will not go into detail of this. You can just read these lines, and some of the theoretical one or two papers sir has also shared, which I think are in good. You should definitely go and read those papers as well. Okay, so now we'll see X inactivation. So we have seen that for the X inactivation, there is a X chromosome inactivation center, which is known as XIC. So what is that? X chromosome inactivation center. It is basically, this is your this XIC means what? It means that this is your X chromosome, and within this X chromosome there is a locus where zist is present and T6 is present and some promoters and some other factors are present, and overall this locus is known as X chromosome inactivation center, which is XIC. So that is what is written over here, that XIC is nothing but X chromosome inactivation center, which is a locus on the X chromosome. Okay, then it contains some non-coding RNA such as ZIST, T6, JPX, FTX, etc. Now, ZIST gene transcribes a long non-coding RNA, which we have already studied, and because it's a long non-coding RNA, therefore it is not translated. Just in word forms it is written. Okay, so is it clear up till here what I'm trying to say?
Ma'am, X inactivation begins from a XIC6 region or
Yes,
6 region.
so basically you can see that so basically X chromosome, when X chromosome inactivation is taking place, so there has to be a region in the chromosome, right? A region from where this ZIST gene is present, a region from where ZIST gene is getting transcribed and RNA is produced. So that particular region from where this ZIST gene is produced and transcribed, and ultimately the ZIST RNA will come and it will bind on this ZIST region, and it will recruit some other histones and some other modifiers, so that the overall transcription of this X chromosome is inhibited. So this is the region which is responsible for the X chromosome inactivation, because it has ZIST gene, which will produce RNA, and that RNA will again come and bind to this gene, and the binding of that RNA to gene will recruit some histone proteins, and those histone proteins will repress the transcription and the expression of this whole X chromosome.
Okay,
Is it
madam.
clear? Okay. Now let us see some questions. Histone unique to bar body is H3A, H1B, H2A 1.2 or H4A. Giving 1 minute to think, and then I will switch the screen to see the answers. Okay. Think we have already studied about it. Okay, now we'll switch the screen and let us see what you have answered. Okay. So, Janara has answered C, Kavita has answered C, and Kanchana has also answered C. Okay. The correct answer is C, that is H2A.1. Okay. So, where we have studied about this H2A.1. Let us see. Um, yeah. See, Zist appears to recruit a specific histone isoform H2A.1, which maintains the chromosome in an inactive state. That means the chromosome which is maintained in an inactivated stage has a specific kind of histone which is H2A.1, H2A 1.2. And bar body is what? Bar body is nothing but an inactivated chromosome. That means this inactivated bar body has H2A 1.2. So Vanaja has also answered it correctly. So the answer is C. Just a moment. Baby, हमको पानी दे दो. हाँ. Unmute कर रहे हैं हम, ठीक? Okay. Now next question is, this is just the question that Janara you have asked right now, just now, that X inactivation begins from a region called XIC, T6, Zist or centromere. I'll give 1 minute to think, and then I'll switch the screen. Think what can be the answer. Okay, so now I'm switching the screen. Let us see what you have answered. Okay. So Kanchana has answered A, Vanja A, Kavita A. So yes. So Jahanara, you have not answered. You have only asked this question now. What do you think should be the answer?
Sixth, I have given answer, ma'am.
Acha, you have acha, okay. C, you have said zist. So why you think it is zist? We are asked C. Okay, I think your doubt is still not clear. Okay, so you see where it is. Okay, this is the whole area, X chromosome inactivation center. The main hero of this center is zist. But Zist is the hero, but it needs some side characters also. And what are those side characters? The promoter regions, some other non-coding RNAs, everything. So for the overall effect, of repression, Zist alone cannot function. It needs the help of side characters also. Okay. So the correct answer would be X chromosome inactivation center and not just Zist. Okay,
Okay,
Zist is
madam.
the hero of the movie, but for the complete movie to become a hit, you need other characters also. So this is like a movie. These are other side characters, and Zist is the hero. So you need everything for the inactivation of X chromosome. So the correct
Okay,
answer
madam.
would be A. That is X chromosome inactivation center. Okay. Now
Thank
next
you.
question is, okay. Zist influence the X chromosome randomly, non-randomly, exclusively, or irregularly. Think, little tricky, not much, but yeah, it might look a little tricky. So think and answer. I'll give you 1 minute to think, and then I'll switch the screen. Think, and then I'll switch. Okay. Okay, so I'm switching the screen now. Okay, let us see. So Janara has answered A. Kavita has answered A, Kanchana has answered C, and Vanaja has answered C. So, Kanchana and Vanaja, any of one of you, can you tell me why you have opted C and not A? What was your reason or logic behind choosing C as the correct option? Tell me. Why C? Don't know? Okay. Okay. The correct answer is A, B, C, D. The correct answer is C. Now, why it is C, I'll tell you. So, this X chromosome inactivation, it takes place majority by random. It's a random event. It's a majority. 90% it takes place majority, or you say 95%. Little bit in some few cases, it can be fixed, and those fixed cases are your imprinting events. That means we have studied that the paternal X is inactivated up till the morule epiblast stage, before the epiblast stage. So X chromosome inactivation, it can takes place randomly on, and it can, in some cases it can be imprinting also. That is fine. But how can we say it is an exclusive? What is the meaning of exclusive over here? It means that if there are two X chromosomes, then X, the activity of cyst is in such a way that it will exclusively work on only one of the chromosome and not both of the chromosome at the same time. I mean, it can choose either this or this. But at one time, if it is choosing this, it will not choose the second one. If it is choosing the second one, it will not choose the first one. So its activity is exclusive to only one of the chromosome, whatever it is, first or second, it doesn't matter. So, मतलब ये है कि if this is your x1, this is your x2, the activity which is happening on x1, it will not influence x2. There would be no influence of the activity of x1 over x2. Whatever it is happening on sorry, I'm extremely sorry for this mess. Whatever that is happening on x, it will retain only on one of the x chromosome. It will not be go on other. Like you must, I don't know whether you have studied or not, but what happens is that sometimes what happens that the activity of one can influence the activity of other, but here it doesn't happen. The activity of one of the gene is not influencing the activity of the other. Cyst is completely exclusive in its function. It can either work in a, it can either be random in choosing the x chromosome, or it can be non-random, but it is always exclusive. Is it clear to everybody? This point, is it clear or not?
Yes, ma'am.
Okay, fine. So now we will move to lecture number 5, which is the last lecture, and then we will have some questions and then we'll wind up. Okay. So this okay, this is actually very interesting. So I have not included all of the papers because you should go and you should just read the papers. There is no point of me explaining the gist of the paper because that would be like a spoon feeding. So I've just included one of the images for your reference to tell you, and also a very important good logic also that I wanted to tell you with this. So you can see over here that X chromosome inactivation and RNAi is RNA interference. So this X chromosome inactivation is actually a kind of RNA interference that takes place. That means you have cyst also and you have T6 also. So if RNA interference is taking place, this is the activated X and this is the inactivated S. So you can see if X is activated, that means if this X is expressing, then cyst is repressed over here, obviously. If this X is getting inactivated, that means there is cyst induction and there is the recruitment of all other factors which are responsible for the silencing of this particular X chromosome. This is like a very basic gist that I am telling you. And if T6 is also present, what will happen? T6 has an T6 has an antisense sequence with the cyst. So it will go, it will bind to the cyst. You see, this red one is the cyst and this green one is a T6. So then what will happen? It will form a double-stranded RNA. And this double-stranded RNA, because it is already double-stranded, so this cyst cannot go and pair, and therefore the activation is stopped. This is how T6 controls regulates the expression of cyst long coding RNA, because it binds with cyst and forms a double stranded structure. Why? Because it has an antisense strand to it. This is just a basic. Now you see, in undifferentiated cells there is both. There is cyst also and there is T cyst or T6 also. But in differentiated cells you can see that one of the chromosomes, which is you can see the expression of T6 is increased. That means one of the chromosome is inactivated and the expression of the chromosome is increased. Expression of T6 is increased, and this, sorry, expression of cyst is increased, and this T6 is still very less. You can see first in undifferentiated cells, as we have studied earlier also, that in undifferentiated cells both cyst I'll just mark it for your pen. Yeah. See, this is an undifferentiated cell. In undifferentiated cells, yeah, this is your cyst and this is your T6. Both are present. But as the cell differentiate, one of the X chromosome is inactivated. And the X chromosome inactivation when takes place, then obviously cyst will increase. You can see it is now increased. यहाँ पे the signal is just a point signal is there. You can see here such a strong signal is there. But this T6 is there. Later on, subsequently, what happens? This T6 is completely removed, and only you have this expression of cyst. So this is how it takes place. This experiment is done with the help of fish technique, which is fluorescent in situ hybridization. That means in situ means in the cell, in the intact cells, you are using the technique. You're not isolating the cells and then you are doing the experiment. That is a different altogether lane. I don't want to go into explaining you the technique. I suppose that you have a basic idea about fish. Now, one important thing that I wanted to point out from here is that sir has also mentioned in the class, and I want to point out especially because in my lab we work on that particular thing only. Is okay, fine, whatever. That if this is your nucleus and this is the center of the nucleus and this is the periphery. So, genes which are repressed, those genes are found to be present in the periphery region, and the activated genes are present in the this region. This region has the activated genes. So, you can see that nucleus divides itself into two compartments. Okay, one is repressive compartment, which represses the transcription, and other is permissive compartment, which activates the transcription, and repressive, it decreases the transcription. So this repressive compartment is this one, the peripheral region, and this permissive compartment is this region, activated regions. So it has been shown, it has been found out through studies, what it has been shown, that certain genes, for example, so God, I think there is some technical glitch. Sorry. It has been shown that suppose a gene which is responsible for eye color, and this is a nucleus from, suppose, let's say in a very vague language, eye, and this is a lung. So what do you think a gene for eye color would be present in eye cell? It would be present in the center or in the periphery? Tell me. A gene which codes for eye color.
Center.
Center. And in the lung cells nucleus, where it would be present?
Periphery.
Periphery. So that is what I am trying to tell you, that the expression of genes always, this is very interesting. Expression of genes is can also be determined by their positions. Very interesting. Even if you don't, if you see some genes, you have no idea about their expression level, you don't have any information what is the expression level, but if you see them in the periphery, you can get an idea, because that since it is present in the peripheral region, that means it is repressed in this particular cell, and if that particular gene is found in center, that means it is expressed in the cell. So it is a very interesting topic actually, and in my lab we actually work on this also. We try to see the switch. So we try to see that when B cells is developed, So B cell development takes place through various stages. So specific transcription factors are expressed in specific stages. So we see that if one particular transcription factor is present at the center, in other cell, in another stage, when it is not needed, it goes from center to the periphery. So this is a very new and a very interesting topic, very different. And I would suggest, if you have idea, you can also read about some of the techniques, 3C, 4C, high C. I don't know whether you have idea about these techniques or not. So these are very new upcoming areas of research which tells us about the permissive and the repressive regions and the switching of the genes from the center to the periphery, or to the periphery from the center. Now, why I'm telling you all this is that if you can see this image, you can see if this is your nucleus, right? And you can see that both of them are initially present in the periphery, but the signal from one it goes, because now the one of the X chromosome, it has, from the periphery, this has been moved to the center. And also because this T6 expression is reduced, that's why you cannot see the signal, but you can see here that it is still present in the periphery only because it is repressed. Okay, now this is just the recap. This is just to mm, recollect what we have studied. You can just go through it. I will not be repeating it, or just let us have a very quick look. So, what happens? What we have studied up to now, that females carry two X chromosome and males carry only one X chromosome, giving a double doses. So, therefore, what happens? To balance the X linked expression, females have a mechanism which is known as X chromosome inactivation, where what happens? There is a random silence of one of the maternal X chromosome during early embryogenesis. Okay. Now, during cell differentiation, what happens? Each cell count its number of X chromosome. The X chromosome is randomly selected for inactivation, and then cyst is upregulated, and then there is a gradual chromosome wide silencing. This is what we have studied, nothing different. And then there are two kinds of X chromosome inactivation. One is imprinted and other is random. We have already studied. Imprinted is like it is a fixed, and random is it is not fixed. Obviously, it is random. So, how it is fixed? Because during the early stages of embryogenesis, it is fixed that paternal X chromosome is only going to be silenced up till certain stages. See, always remember, this is only during the early stages of embryogenesis. It is not to complete offspring formation. No, only up till some stages before the epiblast stage, this takes place. After that, it is random. And then random, so we know that any of the X chromosome can be silenced. And this X chromosome inactivation, it takes place from the X chromosome inactivation center, which encodes 5 non-coding RNA: C, ZIST, T6, XITE, REP-A, and GPX. So that is what I was telling you, that ZIST is the hero and these are the side characters. But for the complete inactivation, all of them are needed. Okay, so that is why it is written over here that depletion of any of the factors, whether it is T6, Z, Rep, A, GPX, it will lead to aberrant number of active or inactive cells. Okay, this is important. Now, in summary, what we can say, that X chromosome silencing takes place through spatio and temporal compartmentalization. So I have not included the papers. You can go and you should definitely go and read the papers and some of the diagrams given. But I have told you the gist of it, that how the compartmentalization helps in the silencing. This is the compartment. First compartment is the central compartment. Other compartment is the peripheral compartments. So all the genes that are silenced, those genes are present in the periphery. Okay. Then in perinuclear space, X chromosome inactivation and other heterochromatin could replicate. Yes, in the perinuclear, matlab in the periphery region. So cyst expression targets the okay, cyst deletion, loss of heterochromatin, transcription, yes. So if cyst is deleted, what will happen? There would be loss of the heterochromatin region and the transcription would be upregulated. Okay. Now we'll come to some of the questions. Okay. First question is, proposed mechanism of T6 and cyst interaction is isolation, masking, competition, or euchromatinization. This is given in one of the papers, I guess, but I will explain you anyways. First, you try to answer. I'll give you 1 minute and then I will switch the screen. Think. Okay. Now I'm switching the screen. So, okay. So, I think people have answered. Let me see what they have answered. Okay. Jahanara has answered A. Kavita has answered A. Kanchana has answered C. Vanaja has answered C. Kanchana C. Okay, okay. That was different. Okay, C and D. Okay, different answers are there. Okay, but the correct answer, nobody has answered. Okay, fine, no problem. So the correct answer is masking. Now, for this, I would suggest you to go and read the this thing papers. But I can just tell you that what happens is that this is your cyst and this is your T6. Now, this is the antisense portion of cyst. So what happens that this T6 will bind with cyst and it will form a double stranded RNA. Okay, and since it will form a double stranded RNA, therefore the effect of cyst would be masked. Why? Because cyst has to be in the single stranded form so that it can go and it can bind to the cyst gene. If it is already bounded with T6, there is no space for complementary base pairing and therefore it cannot go and bind to its own gene. So competition, there is no competition over here. Why there is no competition? It is not like that both cyst is going and T6 is going and whoever reaches the gene first wins the race. No, it is not like this. There is no competition over here that is happening. What it is doing is that cyst is T6 is pairing with cyst. So competition cannot be the case because it is not like that both have to reach fast to secure the position and win the race. No, it is not like this. T6 will bind to cyst. Isolation, why you think there is no isolation? Because this double stranded RNA, it is still present. It is not getting isolated from the you see, nothing is happening that this cyst is getting isolated from the cell. You're not removing cyst, you're just making it incompatible because this T6 has bind to cyst. Therefore the effect of cyst is masked because this RNA strand has bound to it. So there is no complementary base pairing that can happen. Therefore the effect of this cyst is masked, and that is why it cannot perform its function. Okay, is it clear to everybody?
Yes, ma'am.
Okay, the next question is, so I think these questions are from the papers, but anyways I will explain you these questions. And oh god, I don't know why this So I think there is some problem. That's why I can see. Oh, now it is not there. Anyways, whatever it is. So these are some of the questions from the papers that they have mentioned. So these are the questions of the previous years that I always discuss with you. So I will try to explain you right now also. And anyways, these questions would be dealt by next week by other of the days. You need not to worry. Your portion would be covered. Okay. So YY1 influence zist and X expression through mediation, competition, neutralization, and suppression. Just if anybody has gone through the lectures, you would be able to answer. If no, it's fine. So I'll just leave this question. The correct answer is A, which is mediation. Okay, but I don't see that I have that much of time, so I have to explain this whole concept to you, so that you can understand that why the mediation is the correct answer. So I will just leave it. I think your next your other TA would definitely explain you this question. If you're not clear, you can ask me, and I can solve your doubt in your next this thing class. So for now, I'm keeping this question on hold. Okay. Your next, your I mean, you have two TAs, right? So that I think she can explain you, or even if she's not explaining, this is your course and this is your responsibility. You should go and you should read it. But the correct answer is mediation, just for your confirmation per se. Okay. Next question. It is just a minute. Yeah. I think now you're able to see the question. Histone deacetylase causes chromosome compaction. True or false? I'll give 1 minute to think, and then I'll switch the screen. I have explained this logic very religiously today, so I hope you are able to answer this question. Okay. So Kanchana has answered A. Kavita has answered true. Kanchana, okay, that was the first one. Okay. So Kanchana has answered two. Janara has answered true, and Kavita has also answered true. So yes, the correct answer is A, that is true. So I have explained you that I don't know why this is coming. Anyways, so I've explained you that if this is your chromatin, if it is very compacted, then what will happen? There is no place for the transcription factors to come and bind, and there is the transcription is inhibited. That is why heterochromatin region, which is the transcriptionally silenced region, it is so much compacted. So methylation, it makes the chromosome more compact, and acetylation, it makes the chromosome less compact. And the opposite of acetylation is deacetylation. That means methylation and deacetylation have same function, similar function. And what is that function? Their goal is to compact the chromosome, to compact it so that transcription is stopped, transcription is inhibited. Okay. So the correct answer is A, which is true. So now you see everything is gone now. I don't know what is happening. Anyways. Now, next question is, calico cats are always males. True or false? Think, and then I'll switch the screen. Calico cats are always males. True or false? Okay, I'm switching the screen. So, Jahanara has answered true. So, what are calico cats? Anybody? What are calico cats? We have studied. We have seen the example. Yeah.
Random
Random. What?
inactivation of
Yeah.
by
So calico,
cysts.
yes. So that means calico cats are the cats in which you can see that there are patches of both orange and black colors. And why there are patches of both orange and black colors? Because there is random inactivation of X. And we have also studied that this kind of this thing orange and black color patches you can only see in females. Why you can only see in females? Because there only you have two X chromosomes. One bears your orange color, other bears your black color. And if the condition is heterozygous, means one is orange and other is one is orange and other is black, whatever symbol you want to give it, that's fine. Then what will happen? Orange should dominate because it is the dominating factor, but because there is random inactivation of X, so some would be orange and some would be black. So these calico cats are always seen in males. True or false? What would be the answer then?
False, ma'am.
Hmm. Okay. The answer would be false because they are seen in
Females.
females. Okay. Next question is, any chromosome can be inactivated by cyst. True or false? Any chromosome can be inactivated by cyst. Okay, I'm switching the screen now. So, Kavita, Yuvaranjani, Janara, Kanchana all have answered correctly, which is false. Why? Because only X chromosome is inactivated by cyst. Because X chromosome only has the X chromosome inactivation center, which contains the cyst. So it is false. Okay. Next question is, oh God, I don't know why this is happening, but yeah, this is the next question. The maternal X chromosome is inactivated during dosage compensation. True or false? Okay, let us see. So, Kanchana has answered true, and Janara has also answered it as true. So, what do you think should be the answer? I want more participation. The maternal chromosome is inactivated. So, this question um is incomplete, I would say. Why? Because we know. No, no, no. Yeah, I think you all are answered. I am confused with some other question. Yeah, it is. It's a very easy question. The maternal chromosome is inactivated during chromosome compensation. True, but so I was thinking of the other question, but I think I have not included that question. Okay, fine. Yeah, the answer is true. Now, next question is, the best genetic mosaicism is observed in which of the following? Tobacco, fruit fly, dragonfly, and cat. So where you can see the mosaicism, mosaic formation. So mosaic means, so orange and black color pattern that you see, that is a mosaic. So where you can see that orange and black color pattern, I have given you all the hints.
In cat, ma'am.
In cat. So the best genetic mosaic model is observed in cats, and we call it as calico cat. C for calico, C for cat, and it is observed specifically in females. Always remember, it is not observed in males. Now, next question is, what changes occurs in the chromosome to make it inactive? We have already studied about this. What changes occur in the chromosome to make it inactive? Methylation, glycosylation, acetylation, phosphorylation. What changes makes
Methylation.
methylation? Correct. Others? I think, okay. I think everybody knows by now. Inactive methylation, inactive. Acetylation, active. Deacetylation, inactive. And
Inactive.
demethylation,
Active.
active. Correct. So now we'll just very quickly in 1 minute we'll just summarize, remind what we have studied. So first we have studied about tmRNA. What are tmRNA? tmRNA is basically a hybrid molecule which is half tRNA and half mRNA. And what it helps? It helps in rescuing the ribosome which are bound to the mRNA which lacks a termination codon, and also it degrades the that protein. So that we have studied the mechanism how it takes place. You can see this is the tmRNA, the black portion is the tRNA part, and the this portion, red portion, is the RNA mRNA part. mRNA part will give us a stop codon, and this tRNA part will give us an alanine, and then it removes the ribosome, it also degrades the protein. Then we come to dosage compensation. We saw that there are different mechanisms in mammals, in C. elegans, in Drosophila. In mammals, there is inactivation of one of the X chromosome. In C. elegans, both of the chromosomes are present, but they are slightly repressed. In Drosophila, this one chromosome which is present in male, it is hyper expressed. So ultimately, this expression is equals to this expression is equals to this. Then we see that the inactivated X chromosome is maintained in the form of bar body, which is a condensed and darkly stained structure. And then we saw that basically the silencing happens with the help of the long non-coding RNA, which is known as ZIST. So what it does, hmm, so ZIST long non-coding RNA, it will go to its own gene, it will recruit some histones, and it will maintain the chromosome in the inactivated state. Okay, and then we studied that this actually happens in a particular region which is known as uh, X chromosome inactivation center. We also studied about calico cat. We can see it is a mosaic hybrid which takes place only in the females because the X chromosome inactivation is random. Okay. Then we have also studied that X chromosome activation, if it is random, it can also be what? It can also be fixed, and that fixed X chromosome inactivation, it is known as imprinting. Here we have studied that fixed X chromosome inactivation, that is your imprinting, and imprinting of the paternal chromosome takes place up till the epiblast stage. After this, the X chromosome inactivation is random. And then we have saw one of the examples. Here we have we see here you can see correctly, you can see over here that this is the cyst and this is T6. This red one is cyst and this green one is T cyst. They both bind with each other and they form a long double-stranded RNA. So this long double-stranded RNA, it is still present, but because it is double-stranded now, therefore it is unable to perform its function. So this is all we have studied about dosage compensation and X chromosome inactivation. Hope it was clear to you. So now we'll stop the class. We'll meet in the next class then.